citric acid if it has the H on the C minus. There's a hydrolysis reaction here. There's parts of water going in and out of this reaction to make isocitrate. Rearrange the molecule, getting it ready for the next reaction, where CO2 is released. So this is the second molecule of carbon dioxide that is released here in the Krebs cycle. The first carbon dioxide came from where? Pyruvate. That's right. Okay, so here again we have a reduction of an NAD plus associated with this reaction, this decarboxylation reaction. So the product of that is alpha-ketone glutarate. And by the way, we're marking the two carbons that come from the acetyl-CoA here with red or magenta. Right? And those that are already present in this substrate, oxaloacetate, are in blue. So you can follow the fate of those carbon atoms as we go through the cycle. So the next step also involves a decarboxylation and a reduction of NAD+. So we're generating two molecules of carbon dioxide here and two molecules of NADH. So the product of this is succinyl-CoA because CoA, again, coenzyme A, is used in this part of the cycle as well. So coenzyme A is really a way of activating a substrate to make it available for another step in the process. So you see these coenzymes acting at various steps in different processes. This step, again, there's this high energy bond that's formed between CoA and this succinate group. When that's released, that has a lot of energy, so it can be used to make GTP from GDP. So GTP can then be used directly by the cell in several different kinds of processes, or it can be used to make more ATP in this little cycle. So GTP donates its phosphate to ADP to become ATP. Okay? There are a lot of these little phosphate recycling cycles. So you can get another ATP here. All right, and then in the next step, we get a reduction of this oxidizing agent. It's similar to NAD, but it's called FAD because it has a flavin group instead of a nicotinamide group. But otherwise, it is an adenine dinucleotide, and it's just that the flavin group is the hydrogen acceptor, or the electron acceptor. So that becomes FADH2. And then fumarate is made from that. We get a hydrolysis, make phthalate, and we get another molecule of NADH produced in the final step of the cycle. And that leads us back to oxaloacetates. That one's ready again as a substrate for attaching another acetyl group from acetyl-CoA. So you see why this is called a cycle. So with each cycle, in the Krebs cycle, we're getting rid of carbons as carbon dioxide, and we're producing what? ATP. We're producing a molecule of ATP and what? And NADH. So actually there's a bunch of NADH and FADH2. So anyway, these electron carriers are becoming active. Question?
How much of this will we be responsible for knowing in this course?
So don't memorize all the little structures of all these little acids and things like that. Don't memorize the names of the enzymes. Try not to get too involved. I mean, you can. If this helps your studying, you can, trace where every single cycle acts like this. And where's the energy going? And where does it come from? Okay. So remember, the lecture is really about how energy is used, how it's stored, and or how it's taken up, and how it's stored, and then how it's used later. Yeah.
Where is the intermediate step between both of them?
I'm sorry, I didn't hear the question very well.
Where does the intermediate step with the COA take place?
Ah, this one here?
Yeah.
So that takes place in the, I guess just inside the outer membrane of the mitochondria.
The Krebs cycle inside the matrix, right? The Krebs cycle that takes place inside the matrix?
I'm sorry, maybe inside the matrix.
Well, I thought they were doing, I thought the intermediate step was separate.
Okay, now I don't remember. Yeah, so it's, I think it's inside the matrix here. So this would be inside the matrix. This is the inner membrane here. This is the outer membrane of the mitochondria. And so it's being transported first into here. This is where pyruvate dehydrogenase is, and
I just have two questions.
in the back, sorry.
Can the Krebs cycle produce infinite amount of ATPs at once for a long time? Because it seems like it's a cycle. It can go on without...
Keeps on going until you die.
So, do you need any additional anything coming into the cell? Or
Sure. You need, well, so you need those inputs. You need acetyl-CoA coming in all the time, right? In order for this cycle to proceed, you need acetyl-CoA. You need that acetyl group that's coming from pyruvate, and so therefore you need glucose or whatever is producing pyruvate. Yeah.
Yeah, so is it like an evolutionary consequence that ATP is used as the energy currency instead of GTP?
Why is ATP used? I don't really know. It could be that there's a little bit of energy difference there. Maybe ATP is a little higher, has a higher free energy. I'm not sure.
Okay, and then also, if you
Everything, by the way, is a consequence of that.
if you need, you basically need oxalic acid to start the cycle, but then at the same time, oxalic acid is kind of a product of the cycle too.
That's right.
So which one kind of, how did it?
Which comes first, the chicken or the egg?
Yeah, that's interesting. Yeah.
That's a good question. I'm not sure. I don't really know the answer to that question. I assume that these things can be generated in alternative ways. So one exercise that you can do, for example, is to trace the carbons. Where do they come from and where do they go? And how many turns of the cycle does it take for a particular carbon to exit as carbon dioxide? So here's the first carbon of acetyl-CoA, and there's carbon number 2. So I handed out today a reproduction of the cycle here. So just tell me, how many cycles does it take for carbon number 1 to exit as carbon dioxide? How many